Imagine you're carrying this massive stack of pizzas, right? Like
Wow.
a margarita, a meat lovers, a veggie supreme, maybe a Hawaiian.
Sounds like a disaster waiting to happen.
Exactly. You trip, the boxes fall, and all the toppings just scatter across the floor in this massive chaotic pile.
Oh, man.
So how do you mathematically prove which specific piece of pepperoni came from the meat lovers and which, rogue black olive belongs to the veggie supreme?
Right.
In the high-resolution world of spatial biology, that scattered pizza topping is human RNA. And figuring out exactly where it belongs is this million-dollar challenge known as the cell segmentation problem.
It is, yeah. It's a huge issue.
So welcome to today's deep dive. We are unpacking a May 2026 technical report from Nature Genetics. They're introducing this revolutionary computational method called Cellist,
Which is designed to solve this exact mess. I mean, it is arguably one of the most significant bottlenecks in single-cell spatial analysis right now.
for sure. But before we look at how Cellist actually, mathematically sorts those toppings, we have to look at the tools that drop the pizza boxes in the first place.
Right. And that entire field is called Spatial Transcriptomics, or ST. Historically, we would just grind up a tumor and sequence the RNA to see what genes were active.
Like an RNA smoothie.
Exactly. But you lost all the geography. We had no idea where those genes were actually active in the tissue. ST preserves that physical map.
Right, but early versions of this, like 10x Xenium or SlideSec, they gave us the map, but we were essentially looking at the tissue from a commercial airliner.
Yeah, the resolution was pretty low. Or, the field of view was incredibly restricted.
But now, high-resolution ST techniques are zooming all the way down to a subcellular level. And it seems like the field is split into two entirely different engineering camps.
Yes, definitely. So the first camp relies on imaging-based ST. We're talking about platforms like Merfish, StarMap, SeqFish plus Essays, or 10x Xenium. These use pre-designed fluorescent probes that bind to specific RNA sequences directly in the tissue. Then a highly advanced microscope physically images the glowing dots.
So you get incredible single-molecule resolution.
You do. The downside is that you are usually limited to a targeted panel of genes, maybe a few hundred or 1,000, and the physical area you can scan is relatively small.
Right. So what's the second camp?
The second camp approaches it from the exact opposite direction. That's barcoding-based ST like Stereo-seq or SeqScope.
Okay, so instead of searching for specific glowing genes.
Right, they lay down this dense microscopic grid of DNA barcodes on a physical slide. You place the tissue on the slide, and the barcodes act like a sticky net.
Capturing everything.
Exactly, capturing the entire transcriptome, every single gene being expressed across that tissue.
Which is amazing, but that brings us to the root of the cell segmentation problem, right? This phenomenon they call transcript diffusion.
Yeah, this is the killer. To get the RNA out of the cells and onto that sticky barcode net, you have to permeabilize the tissue.
Yeah.
You use chemicals to literally poke holes in the cell membranes.
And I'm guessing the RNA doesn't just drop straight down.
No, it leaks. It diffuses laterally across the slide. So the pristine, sharp boundary of a cell suddenly blurs into this fuzzy, overlapping cloud of molecular data.
Wow. Wait, so we have these ultra-high-definition biological cameras, essentially, but the algorithms tasked with drawing the borders around the cells are basically using a blunt crayon.
That's a great way to put it. The paper mentions that legacy imaging tools, things like Bezo or Sparkle, they use what's called Gaussian priors.
Which means what, exactly?
It means the algorithm assumes the data naturally fits a standard bell curve. In physical 2D space, a Gaussian prior assumes every single cell is a neat, predictable ellipse.
It forces a perfect circle or oval onto the data, even if the real cell looks like a splattered paint drop because of that diffusion.
Exactly. The math just completely breaks down on irregular, messy barcoding data. So then researchers tried the other extreme, unified deep learning methods.
Things like SCS and UCS.
Right. These neural networks are trained to draw strict morphological boundaries based on intense feature extraction. But because they are so aggressively hunting for a physical boundary, they end up scooping up extracellular noise.
Or sweeping in RNA that actually just drifted over from a neighbor.
Exactly. And on top of that, running a heavy neural network on a massive tissue sample requires staggering amounts of memory and runtime.
Okay, so before we get into the solution, let's just do a quick recap for you listening. We've got ST, Spatial Transcriptomics, mapping RNA in space. We have barcoding versus imaging as our two main toolkits.
Right.
But poking holes in the tissue creates transcript diffusion, blurry borders, and the old software tools are either painfully slow or they just assume every cell is perfectly round.
Which they absolutely aren't.
Right, so think about your own field for a second. Have you ever had a tool that worked perfectly in a controlled, pristine lab setting, but just completely fell apart when you applied it to real, messy, unstructured data?
It's universally frustrating.
It really is. So since the blunt crayon methods fail, we need a smarter algorithm. Enter Cellist. How exactly does it reassemble this cellular puzzle? I actually have a mnemonic for you listening to remember how Cellist works.
Oh, I like mnemonics. Let's hear it.
NAP. NAP. Nucleus, Align, Probabilistic Assignment.
Okay, that's actually perfect. So, N. Let's start with nucleus segmentation. Cellist starts by finding the anchors, the nuclei of the cells,
Why start there?
because it gives the algorithm a definitive physical starting point. And Cellist actually offers two different backends for this, depending on what tissue you're looking at.
Oh, interesting.
Yeah, for standard tissues, it uses a traditional watershed algorithm. You can picture the intensity of the image like a topographic map.
Oh, so the bright nuclei are like valleys.
Yes, and the algorithm essentially simulates flooding those valleys with water to figure out where the dividing ridges are between them. It's incredibly efficient. Doesn't even need a GPU.
Okay, but what if you're dealing with a really dense, complex tissue, where the nuclei are just crammed together and overlapping? A simple topographic map won't cut it.
Right. For that, Cellist can switch to CellPose. That's a specialized deep learning model trained specifically on complex cellular morphology.
Got it. Okay, so we have our anchors. That brings us to A in our NAP mnemonic. Align.
Multi-modal registration. This is where so many tools fail. You have two entirely distinct layers of data, right?
The spatial RNA spots and the actual stained image of the tissue.
Exactly. Cellist uses a tool called Spatial to align these modalities. It performs rigid affine transformations.
Rigid affine transformations. Okay, let's unpack that.
It just means it can translate, rotate, and scale the two layers until there is a pixel-perfect overlap. But it does this without locally warping or stretching the biology.
So it preserves the true physical distances between everything.
Yes. And once everything is perfectly aligned, we hit the real genius of Cellist, p-probabilistic assignment.
Right, because now you have a stray RNA spot just floating out in the diffuse space. The algorithm has to decide which anchored nucleus gets to claim it.
Right. And it solves this by minimizing two things simultaneously. First, physical distance. Mathematically, how far is this stray spot from the nucleus?
Makes sense.
And second, expression dissimilarity. Does the RNA profile of this random stray spot biologically match the RNA profile of the nucleus?
Which is based on this huge finding in the paper that nuclear expression and cytoplasmic expression are highly correlated.
Exactly.
Okay, wait. I have to push back on that for a second.
Sure, go ahead.
What if a cell is highly polarized? Like, take a neuron. The axon can extend incredibly far away from the main cell body, the soma.
Right.
The RNA out in the axon synapse is fundamentally different from the RNA being transcribed back in the nucleus. So it doesn't, assuming the nucleus matches the cytoplasm, completely break down there?
That's a totally fair point, and the authors actually acknowledge this limitation directly. Cellist is not currently designed to distinguish intricate organelle-level RNA distribution within a single, highly polarized cell.
Okay, so it has blind spots.
It does. But if we zoom out to the broader tissue context, the vast majority of cells like epithelial cells, immune cells, stromal cells, they exhibit a remarkably high correlation between nuclear and cytoplasmic RNA. For tissue scale mapping, it's an incredibly safe statistical bet.
Okay, fair enough. So how exactly does it calculate both things at once?
It balances them using a hyperparameter called alpha. Alpha is basically a slider. If you crank it one way, the algorithm heavily trusts physical proximity. It assumes RNA didn't float very far.
And if you crank it the other way?
It trusts the genetic matching. It says, hey, this RNA belongs to that cell because it looks exactly like that cell, even if it drifted pretty far across the slide.
But wait, how can you do a genetic match on a single spot of RNA? Isn't a single spot incredibly sparse?
Yes. It's mathematically impossible to correlate a tiny fraction of RNA with a whole nucleus reliably. So Cellist uses an augmentation technique. It basically borrows information from neighboring spots.
Like a neighborhood watch for RNA.
Basically, it looks within a 2.5 micrometer radius. By smoothing the data within that tiny neighborhood, it generates a robust enough profile to run a valid Pearson correlation against the nucleus.
Okay, that is brilliant. Let's do another quick recap. Cellist uses NAP nucleus identification, alignment of the data layers, and probabilistic assignment.
Balancing distance and RNA similarity smoothed out over a 2.5 micrometer radius.
Exactly. So here's a question for you listening. If you were designing this algorithm, how much weight would you give to physical distance versus genetic similarity? Like, which do you naturally trust more in a messy biological environment?
That's the billion-dollar question with the alpha parameter.
It really is. All right, so we have this mathematically elegant system, but an algorithm is only a theoretical equation until you test it against the messy reality of biology. How do we know Cellist actually works?
Well, they ran rigorous benchmarks on a mouse brain Stereo-seq dataset, but they immediately hit a wall. There is no ground truth.
Right, we don't have a magical master key showing exactly where every cell border is in a slice of brain.
No. So they had to use clever proxies to grade the evidence. The first was spatially agnostic random correlation.
Okay, walk me through that.
They took all the individual transcripts that Cellist assigned to a single cell, then, completely ignoring where those transcripts physically sat on the slide, they randomly split them into two piles.
Oh, I see. So if the algorithm actually segmented a single real cell correctly, pile A should genetically match pile B.
Exactly, because theoretically, they originated from the exact same biological source, and Cellist scored the highest on this metric by far.
Wow. And they also used a variance-based purity score.
Yeah. This compares the internal gene variance of the cell boundary Cellist drew to the variance of the surrounding neighborhood.
Basically checking if the inside of the cell is genetically pure compared to the chaotic noise right outside its border.
Right. And Cellist showed minimal transcript contamination, but mathematical purity isn't enough. It has to make the biology clearer.
Biological interpretability. They compared Cellist's output to standard single-cell RNA-seq references.
Yes, and Cellist had the highest concordance and the highest marker specificity, which they measure using log2 fold change.
Log2 fold change. That's essentially taking the logarithm of the ratio of a gene's expression inside the segmented cell versus outside it.
Correct. It proves that Cellist is successfully separating mixed signals. If it says a cluster is a neuron, the RNA inside perfectly matches known neuron markers, separated entirely from the blurry noise of adjacent cells.
Okay, so it's mathematically better and biologically accurate.
Yeah.
But what really caught my eye was the cross-platform scalability, the speed test.
Oh, this is the paradigm shift. So they tested it on a Shake Plus dataset of NIH3T3 cells. Here, they actually did have a manual ground truth.
Oh, perfect.
Cellist beat all the old tools. Bezier, SCS, UCS on intersection over union, or IOU, basically a geometric measure of how perfectly the predicted boundary overlaps the real one.
Right, but then they hit it with the ultimate stress test.
A massive 10x Xenium human melanoma dataset. Huge field of view.
Okay, get this. Running that deep learning tool, SCS, on this dataset took 55 hours. Bezier took 8 hours.
And Cellist.
Cellist processed it in 7 minutes.
7 minutes.
It's not just an incremental update. Going from a 55-hour computing task to something you can run during a coffee break, that fundamentally alters the workflow of clinical trials.
Absolutely. And the output was better, too. SCS completely failed to identify B cells in that melanoma tissue. It couldn't distinguish the keratinocytes. Cellist found them effortlessly.
Okay, recap time. Cellist was validated via random correlation, purity scores, and ground truth IOU. It's biologically accurate and astonishingly fast. 7 minutes versus 55 hours. So think of a time in your own life when a new tool wasn't just, slightly better, but exponentially faster. How did that completely change what you were actually able to achieve?
When you clear a computational bottleneck like that, you can move directly into complex clinical applications, which is exactly what they did next.
Let's take it to the clinic. They looked at non-small cell lung cancer, specifically patients post-neoadjuvant immunotherapy.
Right. They wanted to understand the tumor microenvironment. Why do some patients respond beautifully to immune stimulants while others see zero effect?
Using Cellist, they mapped out 8 conserved spatial ecotypes, basically different neighborhood zones ranging from the tumor core to immune-enriched stroma.
But the most fascinating discovery happened at the tumor-stroma boundary.
The front line of the cancer war.
Exactly. Because Cellist gave them such precise, uncontaminated data, they could isolate distinct niches of macrophages right at this boundary.
And macrophages are incredibly plastic, right? Depending on their chemical state, they can either act as soldiers attacking the tumor or as bouncers protecting it.
Yes. So look at patient ST1. Their boundary was packed with 2 types of macrophages, CXCL9 positive and SPP1 positive.
Now, CXCL9 positive makes the tumor hot, right? It brings in the heavy-duty cytotoxic T cells.
It does. But the SPP1 positive macrophages were physically right alongside them, acting as a chemical wall. They were literally excluding the T cells from actually entering the tumor core.
So the troops arrived at the border, but the bouncers wouldn't let them in.
Exactly. Now contrast that with patient ST5. They also had the CXCL9 positive macrophages, but instead of SPP1 positive, they had SLPI positive macrophages.
And what did that do?
It created a completely cold environment. Dangerously low MHC class II expression, weak cytotoxic signals. It acts like a dampening field. The exact mechanism of SLPI is a totally new mystery to be solved.
That is just incredible. Without accurate cell segmentation, those specific macrophage subtypes would just be a blurred soup of RNA.
You wouldn't be able to see who was interacting with who?
Right. Cellist provides the precise resolution needed to see the literal battle lines being drawn in lung cancer.
It's game-changing.
Let's do our final mini recap. Cellist mapped the lung cancer microenvironment, revealing how specific macrophage combos like CXCL9 with SPP1 or SLPI dictate whether an immune response can actually penetrate a tumor.
Yep.
So, for you listening, if we can now computationally identify the exact cells acting as bouncers, keeping the immune system out, how might that completely change the way we design future cancer drugs?
It transitions the whole field from just observation to targeted mechanism, but we do have to impartially review the limitations here. Cellist is powerful, but it has boundaries.
Right. It prioritizes expression recovery over exact morphological boundaries.
Exactly. If the RNA has diffused widely, Cellist will theoretically assign it to the right cell and recover the molecular profile, but it won't draw the perfect physical shape of the original cell membrane.
It's prioritizing the ingredients over the exact shape of the pizza bomb.
Yes, perfect callback. It also applies its probabilistic rules globally. It cannot yet dynamically adjust its hyperparameter, that alpha slider, based on the specific cell type it is looking at.
Like a macrophage and a fibroblast might diffuse differently, but Cellist treats them the same mechanically.
Currently, yes.
Well, Cellist turns blurry spatial transcriptomics into high-fidelity maps, but it leaves me with this really provocative thought to mull over.
What's that?
If we are moving toward a world where algorithms can computationally reassign diffuse molecules back to their origin with such incredible probability, are we eventually going to just stop trying to perfect the physical tissue chemistry in the wet lab? Like, will we rely entirely on AI and math to clean up our messy biological data post-capture?
That is a massive question. Where is the line between biological measurement and computational reconstruction?
Right. I want you listening to ponder that. How could multi-modal data integration combining noisy physical captures with highly probable algorithmic reconstruction be applied to your own industry? Could you stop trying to build a better microscope and just write a smarter algorithm?
It definitely changes how you look at messy data.
It really does. That's all for today's deep dive. Keep questioning the boundaries, and we'll see you next time.